Hi, my name is Adrian, and in this tutorial video I'm going to show you how to use the ExIR function from the R package influential for the identification and prioritization of the features based on purely experimental data. So let's start. First, load the influential R package, and we prepare actually some sample data to run the ExIR model so that you can apply the ExIR model on your own original and experimental data. And for the sake of this tutorial video, suppose that we are trying to run the ExIR model on a data set that corresponds to a time course experiment that includes 3 different time points, and we try to generate some sample data and some differential and regression data and the experimental data itself. So let's start. We first generate a character vector of gene names that includes a list of 1000 genes with different names, and if you have a look at the head of the gene names, you see a character vector is generated with different gene names, gene 1, 2, 3, etc. Then we set the seed so that actually the purpose of this seed is for reproducibility, and if you use the same seeds that I'm using for this tutorial video, you can regenerate the same results. So we first generate a differential data frame that corresponds to differential expressions of time point 2 versus time point 1. And this data frame includes differential expression data for 90 genes. And again, we set the seed and get a sample of gene names and set this sample of gene names to the row names of differential expression data we just generated. Then we set the seed again and regenerate another differential expression data frame, which includes differential expression data for 121 genes. And again, we set the gene names of this differential expression data. And also suppose that you have done a regression analysis or time course analysis to see which genes within the data set are significantly altered across all of the time points. And it is not actually mandatory. Providing regression data is optional, and it is up to you. You can have only a case control study and generate only a single differential expression data frame, and that would be enough for running the ExIR model. But in some cases, you might do analysis on a time course experiment, and you will generate different differential expression data, and you may also generate some regression values. So it is possible to input all of them into the ExIR model. But before we proceed, we should again set the gene names of the regression data, and after setting the gene names of the regression data, we should sort of integrate all of these differential data frames and the regression data together to prepare that for inputting into the ExIR model. So for this purpose, you can do this manually, but a function has been provided named diffDataAssembly that you can use for automatically generating the diff data which is required for running the ExIR model. And for this purpose, you need to only input the data frames into the diffDataAssembly function, and it will automatically generate the data. And if you have a look at the top 10 rows of the data frame, you see the first column is diff value 1, and the second column is sig value 1, which correspond to the first data frame we inputted. The third column and fourth column correspond to the second data frame we inputted, and the third data frame, which is the regression data frame, and it included only R² values, and we didn't provide the significance values, for example FTR or adjusted p-values, so that the last column corresponds to the regression values of the third data frame we actually provided. So the diff data frame is provided, and we now need the experimental data. And again, we set the seed, and we generate a random experimental data which includes 1000 genes, the same number of genes that we generated their names in the first place. And this experimental data includes 50 samples, of which 50% correspond to cancer samples and 50% of them are normal samples. And we set the same gene names to actually the genes within this data frame. And as you can see, we have inputted the genes on the columns, and we have actually put the samples on the rows. So we generate the experimental data, and again, if you have a look at the head of this experimental data, let's have a look at this in the view pane. You can see that the samples are on the rows and the genes are on the columns. But it should be noted that the experimental data should be normalized beforehand. So if you are doing a bulk RNA-seq, or you are doing a single cell RNA-seq or microarray or a proteomics or whatever kind of experimental data that you are studying and analyzing, you should adopt appropriate normalization steps before inputting the experimental data into the ExIR model. And also log2 normalization is required and is recommended to bring the distribution of your data closer to a normal distribution. And for this purpose, you would do a log2 transformation, and accordingly we use the log transformation to bring the data closer to normal distribution. And again, if you look at the sample of the experimental data, you can see it is now different from the original experimental data. And the final step would be to add the condition column to the experimental data, and the condition column corresponds to the different types of samples. And for this tutorial video, we know that we have two different sample types, a cancer type and a normal type. So we can assign, for example, letter C to cancer and letter N to normal samples, and the first 25 samples are cancer samples, so we assign the first 25 as cancer samples, and the last 25 samples as normal. So we first make actually our experimental data as a data frame, and we set the condition column as the last column within the experimental data, and we can now run the ExIR model. So let's have a look at our input data again. We first need the table of differential or regression data, which was previously prepared, and as you remember, we generated the diff data using the diff data assembly function, and we need to provide the column indices of differential values in the diff data table. And if you look at the diff data table, the differential values are in the first and third columns. So we set an integer vector of 1 and 3 as the indices of the diff values, and the index of regression value, if you have provided regression data within the diff data frame, which in our case is the last column, column 5. So you set the column index of regression values to 5, and you should also identify the column indices of significance values, and in our case the significance values are on columns 2 and 4. So we set the sig values to 2 and 4, and we also generated our experimental data and normalized that and added the condition columns. So you should set the condition column name based on the name that you provided for generation of your condition column. And again, we set the seed, and this actually would be optional. Similar to regression values, it is not mandatory for running the ExIR model, but sometimes you would like to run the ExIR model on your whole experimental data and providing all of the differential values and running the ExIR model to classify all of the genes and prioritize all of the genes based on their features. But sometimes you would like to provide a desired list of genes or features, and you would like to run the ExIR model based on this list of genes. And this could be, for example, a list of maybe upregulated genes or downregulated genes, or if you are interested in some specific genes, I don't know, maybe 10 or 20 or 50 genes that you have more desire to see which one of these genes or proteins or whatever are more important in the context of drivers, and which ones are more important or more sensitive biomarkers, and which ones could be maybe mediators. You can provide your desired list of features, and in this case we generate a sample of gene names and that correspond to 200 genes. So we set the seed and we generate the sample list of undesired genes, and finally we run the ExIR model. And let's review again. We set the desired list, we set the diff data, and we set the diff value, the column indices. We set the regression value, which was the column index of regression values. We set the sig value, again the column indices of significance values, and we input the experimental data, and finally we set the name of the condition column, and the name of the condition column we have previously assigned this object to the name of the condition column. So we also input the name of the condition column, and we set the seed, and you can also set the verbose to true. so that you can see how the model is going. And then we run the ExIR model. And yes, it is finished and the model is built. We now have the results. So let's have a look at the class of my ExIR, our results. And as you can see, the ExIR results have a ExIR results class, which would be required actually for visualization of the ExIR results based on another function of the R package influential. And let's have a look at the names of our ExIR results. And again, as you can see, ExIR has generated data for 4 different classes of features: the driver table, the D-mediator table or differential expression or abundant mediator table, and non-D-mediator table, and finally the biomarker table. And also let's have a look at the results. So as you can see, this is just the head of driver table, and in the driver table we have a score. The higher the score, the more important the feature is in driving the biological process or disease, and the more the Z-score, again, it is more important for running the biological process. And the rank is actually again based on the score, and the lower the rank, the more important the feature is. And there's also a column named p-value, which indicates this actually statistical significance of these scores, and the p-value has been calculated based on z-scores, and also an adjusted p-value based on the Benjamin and Hochberg method. And also we have a column named type, which indicates that whether this feature is an accelerator driver or whether this is a decelerator driver. And the accelerator drivers are those actually drivers that are upregulated in a process, and the decelerator drivers are those features that are consistently downregulated in a process or disease. And if you look at the head of the biomarker table, we have the same columns and the types are different, but almost the same, corresponding to the same concept. The upregulated biomarkers are those biomarkers that are upregulated in a biological process or disease, and the downregulated biomarkers are those that are downregulated. And if you look at the head of D mediators, we have only score, z-score, rank, p-value, and adjusted p-values, and these differential expressed or abundant mediators don't have any specific type. All of them are mediators, and the non-D mediators again have the same columns similar to D mediators. And that's it. Hope you find it useful and use it for identification of the most important biomarkers and also drivers and mediators within your experimental data without dependence on any external source.